World War II now. I think we are ready to start our presentation here in the room. We have everybody situated, and I hope everybody on our Zoom call are ready for a presentation as well. First, let me introduce myself. I am Ina Zukunova. I am Sequence and Application Specialist at Pacific Biosciences. We are thrilled to be back to University of Florida and tell you about some advances we have in our technology, some new kits, some new protocols, some new possibilities. I will talk about new protocols and new products, a little bit of everything. And after my presentation, I welcome our territory account specialist, Emily. She'll tell you about our new and exciting instruments, which we recently launched. Please don't forget to check in. We have exciting giveaways. You'll have a chance to win $25 gift card to Amazon. So let's talk about how PacBio can advance your research in different applications and different areas. So I will talk a lot about how our technology works, but what's especially important to remember when you talk about Pacific Biosciences, that this is a long-read technology, and long-read means really long. We are talking about up to 50,000 base pairs of inserts with very high accuracy, which can achieve Q50, which means 99.999% accuracy on your read. You can do single molecule resolution, and that's how our technology works. Very uniform coverage, which is not affected by GC content or length of the homopolymer or any tandem repeat. And also, what we added last year is the simultaneous epigenetic detection. I will talk about that more during my presentation. The concept, how it works, is very, very simple. All we do is attach smart bells at the end of our fragments and create circular DNA. As you can see over here on the right, we create a circle, and polymerase just goes over and over and over and over again, reading the same insert multiple times. And then when you create the consensus, you achieve high-fi reads. We don't accept anything to the analysis lower than 99.9% accuracy. And the fragment size is up to 25 kilobases. Very simple concept, elegant, works amazing. If you think about that from an evolutionary point of view, it's been in business for millions of years because that's how bacterial plasmids replicate themselves. They just keep reading. If you look at the accuracy on the hi-fi, comparing with our competitors, you could see that PacBio achieves 99.95% accuracy, which is higher than Illumina or any other competitor. And you could see that peak for that hi-fi reads is very narrow. What it means? It means that all the reads are accurate. We don't say, oh, some of them are hi-fi reads and some of them will fix somehow. We just have accurate reads and we don't need to fix anything about that. And what can you do with that? You can do quite a bit. You can do whole genome sequencing. You can do targeted sequencing. You can do RNA sequencing, look at the complex population. You can look at the variant detection. And be rest assured that whatever you read is what is in your sample. So let's talk a little bit about whole genome sequencing and assembly. What is important for this topic? If you look at that, contiguity is very important. Completeness, can you complete the genome? How many fragments you use to complete that genome? is important. How correct it is. Are you missing some point mutation, some lengths of the homopolymers? Is that high base accuracy? Can you phase allele? And when you have all those reads, how easy it is to apply bioinformatics and get your data. So you need to have computational power. And for hi-fi reads, you need much less of that. It's smaller file sizes. You need to do less corrections. You need to do less comparisons. So it's faster analysis time. And then PacBio hi-fi technology can work with very different size of your sample. So if you work with standard whole genome, human genome, plant genome, it's a standard hi-fi sequencing, and you use that DNA directly. You apply smart bells and you sequence it. But it does not mean that you can work with smaller organisms. If you look at, let's say, insect larvae, smaller organism, you need just 300 nanogram. There's a little bit of amplification to run low DNA input protocol on PacBio and get wonderful results. And you also can work with tiniest organisms, tiniest sample size, and with just 5 nanograms of input with some amplification, you can get fantastic data. Just the same quality of data as you get with the normal input. And what that means? That means does not matter what genome. After running PacBio experiments, you can assemble whole genome, either for small body species, large complex plants, and we have data on sequoia and maize, incredibly complex, large genomes. And what it boils down, you have such strong data that you can assemble your genome in 12 hours. Or you can develop reference genomes, which is one of the strongest applications right now for PacBio. And again, why hi-fi reads? It's not only about how precise we are, it's also about how many errors are introduced into your system with the technology you are using. And on the slide, you can see that PacBio delivers a low error rate for any variant types. On the left, you can see the graph, which shows that structural variants, indels, single nucleotide variants, you can call PacBio as gold standard for any detection. With as little as 15-fold coverage. So what are the biggest challenges when you look at the genome? Genomes are big. So if you look at human genome, it's 3 billion base pairs. Plants very often are larger. It's very complex, very many repeat. And like each project is unique, and you need to overcome so very many different challenges. And when you don't need to worry about technological aspects of that, it's incredibly valuable for understanding that you can just run a few experiments and assemble complete genomes. Does not matter plant or animal or human genome, insect. So let's talk a little bit more about complex regions. If you perform short reads sequencing on the complex regions of the genome, you do detect those elements, let's say transposable elements or repeats, but you cannot, as you can see on the bottom part of the slide, you cannot distinguish them in different haplotypes unless you have a long read sequencing, which tells you which structural element on which haplotype is spread. I think unless I point it on the screen, it does not change the slide. Sorry for the delay. I'm working with the clicker still. When you have accurate and complete genome sequencing, you can develop pangenome. And what is pangenome? There are two types of genes in every genome, core genes and dispensable genes. And it's very well known that core genes are present in all the populations of the species. And dispensable genes, they are those which drive evolution. They change fast. They normally responsible for adaptive traits, for rapid evolution, the adaptation of the species. And when you run your sequencing experiment for many samples in the same species, you can finally detect those core genes and separate them from dispensable genes. And as it shows on this slide, on this paper, you could see that structural variants associated more with dispensable genes. And this way, when you sequence, you tap into the evolutionary process. You understand what the basic of that, and you understand the populational biology better, and those experiments are much easier for you. So in this paper, the authors, they assembled 26 genetically diverse genomes and identified numerous complex variations, and majority of them belong to the variable genes. So methylation. Until long-read sequencing with PacBio, it was a little bit challenging, but last year we launched a protocol and detection system which can detect your methylation changes in native DNA. And you don't need to do anything specific. You just make your library. How it works. Remember that circle. It runs over and over and over at polymerase read basis in PacBio Seminar. And it's all normal until it hits a methylated base. And then polymerase stumbles for longer than with normal incorporation. And it's significant, it's statistically significant. So when you see that polymerase stumbling a little bit, you can easily read it as 5MC. So now, instead of reading 4 bases, ACG, you read 5, including 5MC. And you don't need to do anything different whatsoever. So how it used to work, you just make your whole genome sequencing library and you sequence it. And then you needed to do bisulfite treatment, whole genome library prep, and then sequencing to detect 5MC. With PacBio sequencing, you don't need to do that anymore. You just make one library, one sequencing run. You want methylation data or not, you still have it in your front files. And then you can just analyze that and look at it. And combining with long-read sequencing, it actually opens huge possibilities. I really like this data and this paper, which actually shows significance of that. So in this patient, I do not remember what disease it was. Let's say disease X was associated with 5 kb insert. You can see on the left that purple things So 5 kb insert, it's not very standard. It's a little bit smaller, a little bit larger, but it's always there. And it was known for a while that this insert belongs to that disease. But when you do long-read sequencing with methylation, you also could see that this insert was responsible for hypermethylation down the stream in this particular gene. And hypermethylation changed the gene expression pattern. So without actually being able to resolve haplotypes and looking at each strand and methylation together, nobody would ever guess that this insert is also associated with hypermethylation down the stream. In many cases, it's clinically relevant, is very important. And it's just the same thing with this PEG3 gene in inbred mice. So it's a strain of laboratory mice. And it does not, when you look at it, it looks like it does not have any genetic variation within that strain, unless you look at the methylation. And with methylation, you could see maternal imprinting in PEG3 gene associated with hypermethylation. So methylation is fun, but Emily told me to put that slide multiple times. So everybody who did not sign up yet can still sign up and win some prizes. Let's talk a little bit about my favorite topic, applications of hi-fi sequencing in RNA research. And RNA research, gene expression was my favorite topic since I was a graduate student, and always meant to me a lot because yes, there are genes, there are mutations. If they don't express, they mean nothing. Nothing happens. And when I still was at school, the idea was simple. It was just like one gene, one transcript, one protein, everything clear. And then people discovered isoforms. And like one gene, many isoforms, many proteins. Life gets confusing, but much more interesting. And it has huge implication on what we are doing now and what we are trying to achieve. So when you look at the short read sequencing, oops, go back. Okay, here it goes. Clicker does not respond all the time. So let's say this is your gene, and these are different splice variants, different isoforms of this particular gene. If you do short read sequence, and it's like a jigsaw puzzle, you cannot assemble them into the complete picture. You cannot see that. It's all a little bit confusing. But if you read every single isoform from the beginning to the very end, you get that data. You know which isoforms are present and which isoforms are absent. And how important is that? Apparently, it's very important, and we're still learning about that. If you look at this paper, it's very interesting. It's the isoforms detection in single cell format in ants. You could see that different isoforms are responsible for different functions of the insect. It can be workers, it can be other functions. But when you look at the isoforms also, some of them were misdescribed, and only with looking at the full complete picture or the isoform, some genes could be proper identified, and some isoforms mapped with 10x single cell reading were actually attributed to different function of the insect. And how can it be better? It can be much better. So PacBio developed the protocol for single cell. If you go back mentally to that picture of a circle and the insert which you read, it can go to 25 kilobases. However, normal transcript is about 1.52 kilobases. So if you have just insert with that transcript shorter, you are wasting your sequencing capacity. But if you take, let's say, 15 transcripts and concatenate them together in the long strand, you increase your throughput 15-fold, and you can achieve about 40 million cDNA reads in one cell. What that means, you get more data, you can discover more isoforms, you spend much less money doing your single cell experiment, does not matter. For cancer research, investigating those isoforms with ants or looking at some gene expression of plants. And actually, when you align them, even with a smaller coverage, you still get very accurate results. There's a preprint on the cancer research using single cell mass-seq, now we call it mass-seq technology. What did they discover? So they started their experiments with ovarian cancer. They went through with the normal procedure, with that mass-seq procedure. They did single cell libraries with 10x, concatenated those transcripts, ran them with high-five PacBio sequencing on Sequel 2E system. So they increased throughputs. They got about 12,000 reads per cell and with mass-seq protocol. And what they found that quite a few things stood up. They could do cell type classification out of that data based on isoform coverage. They could discover novel isoforms in the system. They can detect new and known fusions in that particular cancer. And they detected cancer-related expressed mutants. And I'll have more on that in the more cells. So if you compare short-read sequencing and long-read sequencing on that, it's no news to anybody that with short-read sequencing, you can classify cells. You do mixed population and you can say this percentage are T cells, this percentage are natural killers, this percent are macrophages, B cells, and so far, so on. And it was the idea that you cannot do that with long-read sequencing because it does not give you enough depth on the expression data, how genes are expressed. However, with the isoform coverage, you can do that as accurate and even more accurate. You could see that clusters for B cells are even a little bit more tighter, and it's more reliable on the B cell coverage. And you just need to actually turn it around just to compare that. But you could see that it gives you close clusters on the cell type detection with long-read sequencing, this mass-seq. Now let's look at the isoform classes. And there's no comparison with the long-read sequencing, as I told you before. So out of that, you could see that out of 100, forgot the number of the total number of the isoforms detected, but 16% of them, you see this green square, they were novel, not in catalog. They've never been described. And we're talking about cancer, which was sequenced over and over and over again, ovarian cancer. Those are novel in catalog, and some isoforms, like 30% of isoforms were novel, not in catalog, and novel in catalog. This is significant percentage, especially when you talk about something as well described as ovarian cancer. And you could see at the differential expression isoforms in tumor versus non-tumor cell. And there's significant difference in the isoforms, which are expected to be cancer drivers. Also, that's actually super interesting for me, at least. If you look at this and you compare cancer over tumor environment, normal cells, you could see shift transition between two cell styles from epithelial to mesenchymal transition. You could see that shift. So the red is the tumor biopsy and blue is the tumor-free, and you could see the shift over here into the mesothelial cells in the complex cell population. And what's also important, as I say, how do you know if mutation is expressed or not? Unless you have high fidelity sequencing, PacBio sequencing, you cannot say if this is a mutation, if this is a minor allele, or this is something which is artifact of sequencing. If you look at this graph on this publication, not publication yet, preprint, we expect it to be in print in major journal within a couple months. But you could see that you can detect somatic mutations and you can detect minor alleles, and you could see how your tumor evolves, and you could see how it responds to treatment if you do consequential experiments. And it can be leveraged for both germline and somatic mutations. And of course, new fusions. Some genes and their fusions are described very well. For some, they are not, but it's very well known that many fusions are drivers for cancer progression, because the gene gets under promiscuous promoter and it expresses more, and there's a system for that. And if you cannot detect that, you cannot detect that. But here you could see that with short-read sequencing, you could not detect this fusion gene, and the gene expression for TESPA1 looked like it was overexpression of the gene. However, it was not overexpression of the gene itself. It was overexpression of that gene being part of the fusion. So that's the statement in that preprint. They demonstrated that long-read sequencing provides a more complete picture of cancer-specific changes. These findings highlight the manifold advantages and new opportunities that this technology provides to the field of precision oncology, opening the premise of personalized drug prediction and neoantigen detection of cancer vaccines. Oftentimes, there are many publications, you could see that isoforms and fusions are valid biomarker or valid drug target for precision oncology or for prediction of the outcome. And now to something completely different. At the end of my talk, I just wanted to add a little bit about metagenome analysis and 16S sequencing. You can imagine that if you take complex population of microbiome and you sequence that with short-read sequencing, you face the same challenges as you can do with isoform detection or with fusion detection. There are many, many little fragments, and you don't see long lines of sequence. Sometimes you miss some elements and you don't know which that piece belongs to, and sometimes a single nucleotide is differentiate the species. So when you look at the PacBio sequencing, those issues are resolved on metagenome level. And you could see that 97% of sequences assigned to genus level using PacBio where, say, a loop-seq at 87%. And even what's even more cooler, that 99.7% of sequences when you do metagenome sequencing assigned by PacBio assigned to the species level. That's a little example of that. Actually, Max Planck Institute, they decided to compare PacBio sequencing, the short read sequencing, looking at microbiome in salt water in different areas. And they are very big Illumina people. They love their Illumina. So they did 11 samples of Illumina, just threw 3 samples of PacBio. Probably they wanted to prove that they can do as well with the Illumina. However, when they run their sequencing, they were amazed because they could complete that they could see significant increase on high quality mags in their samples by using PacBio. So there was 128 metagenomes assembled with 15 gigabyte data with PacBio. Because they had less samples, they ran less. With Illumina, it was 10 times more data, 10 times more sequencing, and just 2 times more mags with so much more effort in the sequencing. And I think that convinced them that it's a very valuable resource. And actually, contigs, there were long contigs higher than N50 value. So that was valuable for them. And you could see here, actually, when you do metagenome sequencing, when you do full sequencing of the complex population, very often 16S getting sort of noisy and dirty, and you cannot deliver complete data from that. With PacBio, it does not matter. The reads are so long and so accurate that you accurately detect 16S region as well, down to the species detection. On this slide, you could see that HiFi data outperforms CLR data on low abundant species. So that's the thing. It's easy to sequence something which is heavily presented, but when it's low abundant, definitely HiFi reads beat any other technology on that. And also HiFi data produce accurate and complete mags at lower coverage, as I already mentioned to you. So in this experiment, what was done? So the samples were sequenced, and then sequences were distributed to the buckets. You could see that bacteria, eukaryotes, viruses, and only a small percentage of the sequencing reads were unmapped. I believe it was less than 10% of reads which couldn't be mapped in that population, and you could see 90% completeness on that. A little bit I wanted to add at the end, because I understand many people are interested in metagenome studies and microbiome studies. So definitely technology discussed with us if you plan any experiment. So that concludes my presentation on updates on the technology. And if you have any questions, I can answer them now. And Emily still wants you to check in. Yeah. Yes, actually, we have benchmark comparisons with standard protocols like bisulfite conversion, and it's very, very close to that, very accurate. I would say yes. I'll get you that data.
All right, so I know you've seen this a few times, but we really just want to hammer home to check in. So we will be giving away a $25 Amazon gift card digitally. So if you are drawn at the end of the presentation, check in with one of us, and then we'll make sure that we have the correct email address so that it just doesn't go into the void somewhere. But you'll see this again a few more times. I apologize for the repetition. So as Ina mentioned, I am here to talk to you a little bit about some of the new products that PacBio launched at the end of 2022. For those of you who aren't familiar, my name is Emily Reynolds. I am your territory account manager, so feel free to either email me, call Ina. Wyra's here as well. It seems like some of you are familiar with her. But my business card is on the back table there. If you do have any questions and you want to get in touch with PacBio, feel free to reach out to me, or any of us for that matter. So the first product that I want to introduce to you today is called Onzo. So as I mentioned, 2022, end of 2022 specifically, was a very busy time for PacBio. We released 2 new systems and a new kit, which Ina kind of talked about already. So the first of the systems is Onzo. This is our new short read sequencer. Oh, this really doesn't work very well. You're right. So as far as throughput goes, the Onzo sequencer, I'm just going to use the, I'm just going to use the mouse and keyboard. The Onzo sequencer is a mid-throughput short-read sequencer. For a single-end read run, you'll get 400 to 500 million reads. Per paired end, it's about 800 million reads. At launch, we're going to have 3 different kits, a 1x200, a 2x100 paired end, and a 2x150 paired end kit. As of right now, the data is looking at over 90% of bases are at Q40 and above. We are still generating data, as I mentioned. And we plan to release Onzo into existing short-read ecosystems as well as provide it its own ecosystem. So we will have conversion kits available, and I will talk about that in just a second. But the special part about our system, because everybody now has a short-read sequencer, right? The special specifications for ours is that it uses sequencing by binding. So if you're familiar with Illumina, they use sequencing by synthesis. And I'm going to hope that this video works. It wasn't working in practice. I will just walk you through it if it doesn't work here. All right, we're going to assume that this is not working. So basically what sequencing by binding is, it's not going to work. So we'll just go one past that so it doesn't freak out. But basically what sequencing by binding is it separates the interrogation and integration steps of your sequencing run. So with Illumina sequencing, it all happens at one time. You interrogate the base and you incorporate it at the same time, which incurs some molecular scarring, right, on your molecule. But with sequencing by binding, we interrogate, hold that at an optimal condition, under optimal conditions, and then it doesn't covalently bind. We track which base it is, and then after we wash off those reagents and run through with actual bases that are not going to incur molecular scarring, and then it's integrated. So you're actually getting just DNA. You're not getting any leftover hanging molecules off of your initial template strand. So theoretically, you can do native DNA sequencing with this instrument if you had enough starting material. I understand that's not the case for most people, but if you did have that, you could. So as I mentioned, Onzo is going to be released into existing ecosystems as well as supported by a PacBio ecosystem. We have partnered and collaborated with various companies. So we're here to support you all the way from setting up your automation all the way into your secondary and tertiary analysis. So these are some of the companies that we're working with right now. As far as library prep goes, we do have what you need for that as well. We have library conversion kits, so any off-the-shelf library prep kit will be compatible with Onzo. All it will need is our conversion kit, which is a 1-hour PCR, 5 cycles, and then it's ready to go on Onzo. We have some PCR amplification kits, and even with these amplification kits, you're not seeing as high of duplication rates, so that duplication rate sits about 3%. And you don't get any index hopping with the Onzo system, which has been one of those benefits that we like to brag about here. So let's kind of take a look into this higher accuracy model that we're trying to convey to you with this Onzo system. So a lot of people ask, why do I need that higher accuracy? And the answer is not everybody will, but for those people who do, this is going to be a very exciting system for you. So what we're releasing Onzo with the intention of is that we're hoping to push researchers who are looking for that needle in a haystack application, kind of like liquid biopsy research and circulating tumor DNA. So in this example, we are looking at a Sericare ctDNA sample, and we ran it through the Agilent Comprehensive Cancer Panel, and we compared SBB, which is Onzo, to an SBS sequencer. So I'll let you use your imagination with which sequencer that was used, but if you can see here, Onzo is in the pink, and then the other sequencer is in orange. So on the right, this plot here is sensitivity plotted against expected variant percentage. So what you want to see is a high sensitivity at a low variant percentage, right? You want to catch those very rare events. So let's take, for example, this 0.1% expected variant percentage. With Onzo, you get 90% sensitivity, whereas the traditional SBS technology, you're getting about 60%. So then what does that mean in practice? Keep trying to use this and it doesn't work. So let's compare, again, let's go into a little bit more details about these runs. So we compared 6,000x coverage of non-UMI sequencing for sequencing by binding on Onzo, as well as sequencing by synthesis on a SBS sequencer. So what you can see here, again, you have that plot over here, similar to the one that we just saw. We're looking at this NRAS gene, which is a known cancer gene, at a specific position along this gene. This data is SBS. So this is what has been used. This is what's being used, what's currently being used right now. So let's look at this table here. Could you confidently say that there's a variant here with this data? So the wild type is obviously a T here with majority T calls, but you're also seeing all the 3 other bases being called here. So if you were just presented with this data, it would be a little bit iffy, right, to say there's something going on here. It could just be the consequences of the sequencing technology and the inherent error rates of that. So now let's look at SBB data. Oops, sorry. So this is SBB data. Sorry, this is not correctly labeled. This should say SBB. The plot here is still the same, but let's look at this table again. So you're still getting a similar amount, similar proportion of your wild type calls, but you're getting 0 Gs and 0 As. So this, to me at least, gives me a little bit more confidence in going to a conversation about a patient who's looking to see if they have this cancer-causing gene. Potentially there's a variant here, right? Potentially there's something to look into. But we also know that SBS technology is being used right now for this exact application, right? So how are they doing that? They're increasing the coverage and they're using UMIs. So let's give this the fair comparison, right? Let's look at SBS data, how it's being used right now in the field. So we bumped up the coverage to 24,000x coverage, sequencing depth, and we gave SBS the benefit of UMIs. So this is what is currently being used versus still that 6,000x coverage of SBB data. And now this is representing both. You can see here on the left that these curves are a little bit more similar now. So keep in mind that's with the improvement for SBS of the depth of coverage and those UMIs. And then here, let's look at this table a little bit more closely. So the wild type call here is a C, because this is a different position. We're actually not expecting a variant here. This should just be your wild type. And here, you are getting just the wild type with SBB, but you're also getting SBS calls that are A's, G's, T's. So this could be a variant, right? You can't really be certain that you're looking at a very rare event here if you're using SBS. And again, the most exciting part of this for me is that this SBB run is still that 6,000x coverage with 0 UMIs, whereas SBS, you have 4 times the coverage, and you have to bioinformatically tease apart those UMIs after your run. So if you could just not do that and save yourself some time, I'm of the opinion that that might make my life a little bit easier, right? So SBB or Onzo technology is still in the active beta testing phase. We are working with Broad Institute, Corteva, and Weill Cornell, and we will have some more data sets here coming out soon. So for reference, as far as the timeline goes, this was announced at ASHG last year, which is like end of October. And we're planning to actually release the system for it to be in commercial hands, so in labs in the coming months here by like Q2, so in the early spring. Here's some data just for the paired end and single end sequencing benchmarks. So the flow cells will each have 2 individually addressable lanes. Again, I mentioned it earlier, you're seeing about Q40 for 90% of those reads. And I mentioned over 800 million paired end reads and over 400 million single end reads. So here's just some quick data about that. And then, yeah, as I mentioned, the timeline, we're accepting orders soon, and then we'll be shipping in the first half of 2023. So that's all I have about Onzo. Does anybody have any questions about this system before we move on to the next? Perfect, and I promised you would see this again. So, oh, David. Duplication rates, yeah. Right. So what David was asking for those Zoom people is how we see those low duplication rates. It is, as far as I understand, a result of the difference in the sequencing technology itself. And we do, as well as the kits that we're providing with the system itself. So we should have more information about those kits coming out soon, just because it's not commercially available just yet, so we don't have all the information. But as it releases, I'll give you some more of those specific details about that. If you want to, so the SBB technology came from Omniome. So if you're familiar with Omniome Tech, the company Omniome, you can look up their tech and it should have some more background information, some more specific about that. And we can also connect you with an FAS or something if you want to talk more in depth about that. Awesome. All right, so we will move on here to the Revio system. All right, 10 minutes left, so this is perfect. So the other system that we launched at ASHG is called Revio. This is our new long-read high-throughput sequencer. So you guys have a Sequel IIe within the ICBR core. This is a new, similar system. It just has higher throughput with a more affordable run cost. So Ina brought this up and her slide looks very similar to this. Same information, it's going to be the same thing that Sequel IIe provides you with. It's that long read, highly accurate, uniform coverage, native DNA molecules, and you do get epigenetics with that still. So epigenetics is free with every PacBio sequencing run, just the long read sequencing run, just by the nature of how we do that. And we collect that kinetic information with every run, no extra wet lab steps. Ina also kind of mentioned this kind of topic as well. So a lot of firsts were achieved with hi-fi sequencing. We completed the telomere to telomere human genome, finally fully completed it last year. Transcriptomes had never been more accessible, as well as the epigenome itself. So let's get into some of these specifications about the Revio system. So if you're familiar with the Sequel IIe, it had about a quarter of this throughput. So this system has 100 million ZMWs per run, and a ZMW is that nano well where the sequencing actually takes place. So on the Sequel IIe, it was able to house 4 8M smart cells. So that means each smart cell had 8 million ZMWs. So theoretically, you could have 8 million molecules sequencing at the same time if it were a perfect world. And this system can do 100 million per run. So this, and I'll get into a little bit more details about that in a second, but you get 360 gigabases worth of data. Your read length is not at all changed. It's still in that 15 to 18 KB range. The sequencing time was decreased a little bit, so 24 hours instead of 30 hours. I mentioned it already, but you get that methylation information for free, and that 90% bases, 90% of your bases are still Q30 and above. So Revio was really designed to address some of the requests from our customers on a previous sequencing system. And those major requests were throughput or scale, ease of use, compute power, and affordability. And Revio really addresses all 4 of these requests. First and foremost, the scale, so the throughput. Revio has 15 times more throughput than the Sequel IIe system through these various avenues. So the higher density of ZMWs per smart cell, I mentioned that you get 100 million ZMWs per run on a Revio. So that comes from 4 different smart cells that run simultaneously on the instrument. So you get 25 million ZMWs versus 8 million ZMWs per smart cell on the Sequel IIe, which could only run 1 smart cell at a time. And then the shorter runtime equates to a 15 times increase. And if you were to look at this in the perspective of how many human genomes can I sequence with whole genome sequencing in a year, you're looking at 88 versus 1,300. So this is quite a throughput increase. I'm sure not all of you are doing human whole genome sequencing, but for those of you that are doing like plant genomes, other animal genomes, this is definitely something to look into because it will bump up your throughput a little bit more here. And I mentioned so that you could run 4 smart cells at once on Revio, right? And all 4 of those smart cells don't have to be the same project. So you could have one person running a human whole genome sequencing sample, and you could do it on 1 smart cell, get a whole human genome. You could do a couple tumor samples. You could also do a mass-seq protocol. So Ina talked about mass-seq earlier about that multiplex single-cell DNA sequencing. You could do one of those on here as well. So it's really flexible. And to address some of that flexibility as well, we wanted to give users the ability to run this instrument 24/7. So for some of those bigger users like a Corteva who sequences all the time, you can imagine that having the instrument be down for any amount of time, like while you're loading instrument or loading your next run, it can kind of wear on your time and your people, right? But if you could run it 24/7, if you had the manpower to run it 24/7, it would really increase your output. So you can actually load consumables as this is running, and it never has to take a break. But we also tried to make it a little bit easier to use and friendlier to approach in general by removing the need for nitrogen. So Sequel IIe system required an external nitrogen supply. We reformatted the smart cell so you actually no longer need nitrogen. And then we consolidated the consumables a bit here. So all 5 of these consumables used to be individual parts on a part list that you would need for a run. That's all included in this one consumable for Revio. So that won't mean a lot to most of you, but for those of you that actually run PacBio and have run PacBio, this will hopefully be good news to you. And then as far as increased compute power, because if you have an increased amount of data generated, of course you need to be able to bump up your compute power so you can actually process that data, right? So the Revio system actually has a 20 times greater compute power than the Sequel IIe. That's through inclusion with NVIDIA GPUs, which was fun for me as someone who, like, I'm a computer building kind of nerd. So that was really exciting for me because NVIDIA is a great company and they have a lot of great technology out there. This is really helping us with our data processing. And then we partnered with Google Health to develop this deep consensus AI tool, which is for data processing. It'll help with base calling, methylation calling, all of these things. And we also created 50% smaller data files. So with more data, it would take a great amount of time to transfer all of that data or process all of that data on instrument, or if you're doing it off instrument, that would take forever. So we wanted to make it easier and faster. That was the hope with Revio, and I really believe that we addressed that. And then for the last part of the equation that everyone asked about, and this is probably everyone's favorite topic, affordability. So for $1,000 or just over $1,000, if you're including library prep, you can sequence. So again, sorry, we're doing this in whole human genome sequencing equivalents. So you could sequence a 30x human whole genome sequencing sample for $1,000, or $995 for just the sequencing consumables, and then library prep is about a little under $100. So for reference, on the Sequel IIe, you would have to spend $3,700 at least to get to that same amount of data. So this is a third of the cost and 3 times the amount of data. So we kind of went both ways there. Here's some data just about some of those initial runs on Revio. So as you can see here, you're getting more data yield, more hi-fi yield with a similar base quality scores. Read length is still about the same. These data sets are available online. So if you are a bioinformatician and you want to look at what the Revio data actually looks like, feel free to reach out to me, or you can just go to the product page. It's listed there, but I'm happy to send you the link if you want to play around with that as well. And then as far as the data characteristics, we want to look into that a little bit more. So that read length, still the same 15-ish KB. Read quality, still massively around this Q30, Q40 range. And when we say that base quality is Q30 and above, we really mean the majority is above Q30, like into the Q40 range. So we're still preserving that high accuracy of the PacBio reads, and we're still preserving that methylation call. So this is a comparison between the Revio methylation calls and the SQL2e methylation calls. They're virtually identical. So you're not losing out on any kind of methylation information from Revio versus SQL2e. If anything, you're just getting more data because you can just get more data in general with Revio. So you're not losing out. Of course, SQL2e still produces this for you, so you can still get this methylation data right now if you were to sequence with the ICBR core on their SQL2e system. And then as far as application performances go and what we've looked at previously, so this is from that Precision Truth FDA challenge that Ina presented earlier. So this is taking that SQL2e data and comparing it with this Revio data. So Revio is in pink, SQL2e is in gray. Very similar across the board. Just want to hammer that home that you're not losing any accuracy. Everything is the same. Your N50s are largely looking pretty good here. So you're not sacrificing anything with this increase in throughput. So in just a basic summary, we increased the scale. We tried to make it a little bit easier and friendlier to use. We bumped up that compute power and also tried to make it a little bit more affordable. So that is Revio in a nutshell. And I'm sorry I went over, oh, that clock is a little bit fast. We're just at 12:59. So if you have any questions, feel free to ask them. If you have to go, that's totally fine. Go ahead and scamper out. But if you want to ask us any questions, we'll be here for a little while. And thank you so much for coming. We really appreciate it. But don't go anywhere just yet, actually, because we do have to raffle off the gift card. Now soon? Okay, perfect. Can you check in with Wyra and make sure that we have your correct email address? Awesome. Thank you all. Yeah, thank you. Know what we do with this.